All right. So we continue. Good morning, good afternoon, dear viewers, friends, and colleagues. So this is our second session with Dr. Rahul Misra, who is joining us from Toronto, Canada. And the conversation is about the analytical techniques that analyze particle size and also those that are used for drug design, for drug formulation, for testing formulation. So without further ado, Dr. Rahul, floor is yours. Please continue.
Thanks, Marina. Thanks for joining again. Okay, I'll bring my presentation. Okay, could you see the slides?
It's coming. Yes. Yes, I
Okay.
see the slides, maybe full size, and then we should be okay.
Yep. Okay,
Good.
so
All good.
just to give people a connection. So last we discussed about the microtrack sync, which was kind of a bridge between microflow imaging and the master sizer, which is a light diffraction based technique. And we could able to not only measure the particle size, but also we can see the morphology of the particles. So that's why, and it's pretty broad in the range of zero point zero 0.01 to 400 microns. So I really like this technique, and it can be very powerful for resolving complex formulations. So switching gears here. So till now, what we have seen is all optical technique, light scattering, light diffraction based. And if you can recall from the flow chart I've shown that there could be a technique which is not working on optical properties, but on electrical impedance. And this technique is called tunable resistive pulse sensing. This is another new technology in the field of particle sizing, and it's been used in different fields, not only just in pharmaceuticals, but also in other areas where people are dealing with particles. And this technique is very powerful in a way that it can resolve particles all the way from 40 nanometer to 20 microns. So you have a pretty broad range to work with. And the way this technique work is also is not an ensemble technique. It's particle by particle approach. So it's basically interrogating each and every particle in the formulation as they are passing through the pores. So if you see this cross thing is a we call this as a nanopore, and this has
Mm.
a small pore in it, which could be the size of 1 microns. Uh, and this size can be tunable. That's why the technique is called tunable. And how it's tunable, when you mount this on a lever, this lever stretches this membrane, or depending, it can go narrower, it can stretch it. So this pore size can be tunable.
Mhm.
And on the top, you load your sample, and you apply electrical voltage around this nanopore. So as these particles are flowing through this pore, there is already established current through the electrodes, and as they are passing through it, they are obstructing, or they're causing a resistance to this baseline current. And bigger the particles, larger the resistance. So this signal, which you can see, sorry. So these signal which you're seeing is basically the electrical resistance to the baseline current. So the larger this signal, the larger the particle size is. So we can understand a little bit more in this schematic. The schematic is from ISON Exoid application notes, and this is how this instrument look like. And they call this instrument as ISON Exoid. ISON is the company, and the name of the instrument is Exoid. So imagine this is your nanopore, and these your particles are in the electrolyte solution, and this nanopore has a baseline current through the electrodes. And as the particles are flowing through the pressurized channel, this you see these kind of signals, and this larger signal is the larger the particle, and that's how you resolve the particles in your formulation. But this is like each and every particle passing through is giving this kind of signals. That's why it's called particle by particle approach. uh, in a technical terms, they call this signals as blockade magnitude. And Brockway magnitude is directly proportional to the volume of each particle, which is you're measuring the absolute size of the particle. Okay. And as I mentioned, it has a wide range from 40 nanometer to 20 microns. You can measure size, concentration, and charge information from this technique to enable nanopores to facilitate measurements of mixed population. So if you have populations of different size, you can change the size of nanopore. You can use the appropriate size, and every nanopore has a certain range of the particles to work with. Okay.
Just
Now,
yeah,
sorry.
just a quick question. So the hydrodynamic radius basically is not a concern here, right? It's really
Exactly. Yeah.
what the
Yeah.
size of the particle is. Because you
Yeah.
mentioned hydrodynamic, right? For viewers, also just remind it's about DLS. And
Yeah.
I think, Rahul, you mentioned that the size will be different in DLS and by other techniques. And I
Yeah.
remember
It can be
distinctly,
slightly over
yeah,
in
yeah.
size.
I remember
So
when you compare with electron microscopy sizing for the
yeah.
same material, the difference was about 10 nanometers. We actually significant if you can
Yeah.
skew your calculation towards a larger size. Sorry for interruption.
Exactly.
Just
And
want to
yeah.
highlight. Yeah.
Yeah, no, absolutely. Thank you for bringing that thing. And yeah, and that's why in my flowchart slide deck, I mentioned about the different techniques, challenges and opportunities. So there are different opportunities using different techniques, but they also come with certain challenges. And that's
Mhm.
why it's important to highlight this here. No technique is perfect, but depends what can solve your problem. Um, so heading to the another new technique is AF4 mols, and it's called as asymmetric field flow fractionation. This is kind of it can sort out the issues we see with the SEC. So size exclusion chromatography is a very common technique used in industry and academics and in different fields, not just in pharma, to separate the particles of different size and as they're flowing through the column, and this column is packed with beads and of different size. So some particles they spend time, the larger particles they spend less time in the column, while the smaller particles as they pass through the bead they spend more time and they elute out later. But what happens, it has been found that some particles have non-specific interactions with the column beads, and the particles are not large in size, but they are spending more time in the column because of the non-specific interactions with the column. So not necessarily they are bigger in size, they're smaller in size, but because they're having interactions with the column, so the separation is not accurate. Also, sometimes some particles are they are falling apart in the system, and they are so fragile. So it's a SEC could not be a useful technique to separate those aggregates or those kind of formulations, and that's where AF4 mols comes into the picture. Here, there is no interaction with the column. This you can imagine this channel. I will call this as a channel and not a column. This channel has a semi-permeable membrane, and on the top, you're injecting your sample, which has mixed of different populations. So you see the larger green particles and the smaller purple color particles, and there is a flow moving in this direction. So as the particles come down through injection and with the flow in this direction, there's a cross flow happening, and there's also a diffusion of particle happening. So smaller particles will diffuse up and the larger particles will go down. And because of this cross flow, they will try to separate themselves in terms of the size. So you have a cross flow, you have a diffusion, and then you have a parabolic channel flow. So in this direction, and that's how the separation happens. But there's no interaction happening with the membrane. There's no interaction happening. There's no. This is not a like a. It's not a shear stress happening on the particles. So it's very powerful technique in that way that it can resolve the populations which are very fragile in structure. And it can work from 1 kilodalton to 100 kilodalton. So this is another schematic from Wyatt, and this explains better what's happening inside this channel. So you see the particles larger and bigger as they're injected, and due to the plug flow, it's just moving in this direction. But as they're moving in this direction, there's also the diffusion happening, that the smaller particles will diffuse faster compared to the larger particles, right? So that's how they get eluted or separated. So as I mentioned, it has a low shear forces. You can have a better sample recovery, and the fraction collection is easy. We can work on a larger size range from 1 nanometer to 50 microns. The only limitation is the flow membrane, the molecular weight cutoff, and the sample concentration needs to be optimized. As it fractionates the samples based on their size, particles with same hydrodynamic size but with different morphologies or surface molecules or other biophysical properties cannot be separated from the AF4 alone. So you may need an additional technique. Sample can be lost if there's an interaction with the membrane. So that's another thing we need to check while using this technique. Okay, so till now, we covered all the particle sizing technique. So if you can recall from my flowchart, I talked about the stability of the vaccine formulation. And in that stability, I talked about the colloidal stability and conformational stability. So colloidal stability was coming from the particles being suspended in the solution, not aggregating. And we can use these particle sizing techniques to tell if the particles are aggregating or not, what are the size. So now we going to talk about the conformational stability. And for conformational stability, DSC, which is differential scanning colorimetry, is the gold standard technique used in industry, pharmaceutical industry. But we also have another technique. So that technique works on a different principle, but we have a new technique which is called nano DSF or DSF, which is differential scanning fluorimetry. They both give the same, like similar information, but they work on a different principles. So DSC works on change in the heat capacity when during the thermal ramp or during the thermal unfolding of a protein. So when a protein is unfolded, so the energy required from one state to another, from a folded state to unfolded state, is basically measured as a heat measure of a heat capacity by DSC. While DSF works in the change in intrinsic fluorescence. So we know that proteins have tryptophan and tyrosine residues, and they have intrinsic fluorescence. So as when I'm heating a protein and they are being thermally unfolded, these tryptophan and tyrosine residues are exposed. And when they're exposed, there's a change in fluorescence happens. And this is what this technique measures. So change in intrinsic fluorescence of tryptophan and tyrosine residues with respect to change in temperature. And there's another advanced version of nano DSF. So there are two versions. There is Prometheus, nano DSF Prometheus and nano DSF Panta. So nano DSF Prometheus just measures your thermal transitions or thermal unfoldings. We can also call this as melting temperatures. And also it has inbuilt back reflection optic system, which can also measure the change in size while the thermal unfolding is happening. So what it does is like, so in this one, on the left schematic, you see there's a folded protein, and as it's being unfolded, the tryptophan residues are exposed. So from folded to unfolded state, the tryptophan fluorescence increased, and this is what measured by as a measure of melting temperature. Now the colloidal stability aspect. So, for example, your proteins. This is called a stable colloidal solution as your vaccine candidates are fully suspended in the solution. And then the unstable solution is where they start aggregating or settling down or crashing out of the solution. So this inbuilt reflection, inbuilt scattering system, what it does is when there's no aggregation. So what's happening, what we are trying to do here is like, imagine if I'm heating my vaccine formulation or there's a temperature change happening during the transport or something. The question we can answer through this technique is my protein both conformationally and colloidally stable? What if I increase the temperature from 25 to 50 degrees, are the formulations start aggregating. So you can tell from this technique that as the formulation is being heated, you see this is where the no aggregation happening. So when you expose the light, the laser light, the backscattered light is being detected by the detector. When there's an aggregation happening, it will have more scattering of the light. So here you can see with the increase in temperature, these are all non-aggregated vaccine candidates. But as the temperature is increasing, they're all aggregated, right? So with that, there will be more scattering of the light.
Mm.
So with so here again, this figure explains this better. So see, these are all non-aggregated. With increase in temperature, they start aggregating, and as they start aggregating, you can see the increase in the scattered light, or the you can also represent as an aggregation. So this is very powerful technique in terms of telling um, how the protein is unfolding with increasing temperature, and at the same time, are they aggregating or not?
Yes, and you can see it distinctively, whereas like in DSC, we can only guess by the fact that the baseline changes significantly after melting, or
Yeah,
at the melting you see very sharp peak and it looks very, how to say, it's not smooth, it's not bell shaped, but kind of abrupt. You
yeah,
can only state from experience, yes, that's what happens, but here you have basically, how to say, not numeric, but digital representation
yeah,
of
yep,
exactly.
yep,
So right, so
exactly.
that's a big difference
Yeah,
with decision making.
yeah. So because we can study the aggregation through those light scattering techniques which I mentioned before, or particle sizing techniques, but those are at a certain temperature where your formulation is stored. For example, I've stored the formulation at 5 degrees for for a week, and I want to see the formulation is being stable or not. But this is where we're talking about in terms of transportation. This technique is very helpful to tell what will happen if you transport your formulation and if there's any temperature excursion happens, right? So is your formulation is stable between 25 to 30 degrees, or if for certain reasons the formulation has been exposed to a higher temperatures, is it still stable,
Yes, you
right?
can have a predictive, basically know where
Yeah,
you're heading, right? Like a predictive technology.
exactly.
For sure.
So this is a little bit more in depth about this technique, how this instrument look like, how it works. So you see, I got this images from the Nanotemper Technologies application note, and this is the tech company which make this instrument. So this is how it looks like, and it opens up as it is a drawer which opens, and there's these small capillaries which sits on this slide, and each of these capillaries can be loaded with different samples, and but everything is exposed to the same temperature ramp. And you see that why we call this as nano DSF is not because we are working in the nano volume, but because the volume of the sample is so low, it's just 10 microliters. And then as you load these samples, it just goes in, and the whole thing is being exposed from temperature ramp, whatever ramp you set. It can goes from 15 degrees to 95 degrees, and the rate of the ramp can also be controlled, whether it's 1 degree per minute or 2 degrees per minute. And then at the same time, this fluorescence detector just moves on each and every capillary and detects the change in fluorescence. Um, and then this is as I mentioned, it can do the thermal unfolding. It can also do measure the thermal aggregation. So through thermal unfolding, we get the information about the melting temperatures, which is what the fluorescence detector is detecting. And the thermal aggregation part is the light scattering. So there's another detector which goes around and see how much light is being scattered from each and every capillary, and through that we get the information about the thermal aggregation. Okay. So that's another uh, slide to talk a little bit more about the capability of this instrument. So if you remember, um, I talked about DLS, the dynamic light scattering. So this component is already inbuilt in the system. So this system can work as both nano DSF and DLS. Like the microtrack sync was working as a master sizer and MFI. The upgraded version of nano DSF can give you more information about DLS and nano DSF. So not just at different temperatures, this instrument can also give you the size of the particle at a certain temperature. So, example, you want to know what would be the particle size at 50 degrees of my formulation. You set the temperature 50 degree and you could get the size of the particles. So that's why it's pretty powerful technique, which combines the modality of two different technologies. So I'm a little bit switching the gears now here. We talked a lot about development of vaccine formulation, characterizing, studying about the stability, seeing the different techniques where we can use. So those are like kind of your tools we can use for during the development phase of the vaccine formulations. Now let's talk a little bit about the transport and delivery, because if you remember, I mentioned these are three different phases, and the transport and delivery is the another two important phase in sustainable vaccine formulation. And when we talk about transport and delivery, is the first factor comes is the longer shelf life. And why we need longer shelf life? It reduces the cost. Packaging, how are we going to package this, the vaccine formulation which we have developed? Are we going to package in pre-filled syringes? Are we going to package in glass vials? And what would be the environmental impact of these packaging? How are we storing it? Are we have to monitor the temperature? Do we need to maintain the cold chain integrity? And are there any data-driven AI models or predictive models which can make this whole process sustainable? Do we need to train the clinicians? Is there any specific training which we need to provide to the nurse practitioners, to the clinicians to administer these specific vaccine formulations? So all these factors are very important to design a sustainable vaccine formulation. So we're gonna take a little bit more into depth of this. I found this schematic from this paper from Chen et al. It's a recently published paper from last year in Journal of Pharmaceutical Sciences. And they have shown the different phases and what are the factors which are important in each and every phase of the vaccine manufacturing. Say, example, first is the raw material supplier. To make a vaccine, we need some raw materials, such as could be the carrier, the antibiotics, the surfactants, the detergents, the adjuvants. And this goes into the vaccine manufacturing, and then the vaccines are made. It could be different types. It can be recombinant vaccines. It can be whole protein, the protein antigens, can be whole virus. And then once the vaccine is developed, it goes to vaccine warehouse. That's where the storage thing happens. And then from vaccine warehouse, it goes to the health center. And from health center, it goes to the immunization venue. And in the health center, we have certain social responsibilities that, okay, what are the unmet demands? What are the deprivation factor? Is the accessibility rating? And the employment impact factor as well. And at the end, it goes to the patients. But in entire process, there are three important factors, such as what kind of waste are we generating? Is it the waste related to vaccine packaging materials, syringes, needles, empty vials, vaccine vials, the dry ice where we storing the waste? And during this whole process, the transportation is an important factor. We are storing, what temperatures are we storing from -50 to +8 degrees? Uh, how are we transporting? Is will be refrigerated vans, cool boxes. So these things are very important. And then the waste which are generated, are we segregating it, sanitizing it, recycling it? So this gives a whole picture in a broader perspective. And based on this whole scenario, they came up with the vaccine cold chain network design model. They have developed a model by considering all these factors. Um, a vaccine cold chain network involves, which I've really found very interesting. I thought to share it here. Of course, it was very mathematical model, but it could be very useful for your viewers to go and
Yes.
to refer this paper and get a deeper
Mm-hmm.
understanding. But I try to bring something very basic about this model and what they are trying to do and what they come up with. Uh, so this vaccine cold chain network involves raw material suppliers, which is is designated as S, manufacturing plants. From manufacturing plants, it goes to the warehouses, and these are different transportation modalities. And from warehouses, it goes to the recipient zones. But in all this whole process, there are economical, environmental, social, resilience, performance metrics involved, which they have accounted in their network model. And the goal was to optimize this whole network, including the location of facilities, including vaccine raw material suppliers, the plants, and the warehouses, the quantity of vaccine transported between the facilities, as well as allocation planning of recipients with respect to minimizing the economic consumption and carbon emission while maximizing the social responsibility and resilience. So the way they did it, they have established a multi-objective vaccine cold chain network design model. And again, the aim was to reduce the economic consumption, carbon emission, while maximizing social responsibility and resilience. To enhance the waste management, they integrated it as an economic metric into the economic objective. And the data-driven machine learning method, which is they call it as WDRO, which is Wasserstein distributionally robust optimization method, was used. And this is kind of a method which accounts for the uncertainty in addition to the conventional stochastic programming and robust optimization. And based on this model, they came up with the CEI factor. And as you see, there are like all these factors are involved, and these blue points, it rates each and every parameters, that which parameters involved the maximum carbon emission. So the data indicated that the transportation has the highest environmental impact, while vaccination, raw material supply, and the waste management show lower values. So consequently, reducing carbon emission during the cold chain transportation is essential for promoting environmental sustainability throughout the vaccine life cycle. So if they can able to efficiently manage the transportation, the whole cycle would be less carbon emission. So, and they also um, gave the liberty to the users while using this model, that they can, because this every life cycle, this model could be different. For some setups, this that maybe transportation is not required, you're actually producing at the site of vaccination itself. So this model gives that liberty to the user to rate their factors based on the based on the time they're investing, or so I found this very interesting, and could be useful for the users to go and refer this paper and read more about this model and use in their knowledges.
Thank you so much, Rahul. It's really great. And I think it resonates with the other speakers. I think we had, not recently, maybe a few weeks ago, Ricardo Martinez Villegas. You probably know him from Sanofi time. He's now heading his own company called TerraBiome. So part of his talk was dedicated where you can cut, and apparently it's a shorter logistics, shorter
Yeah.
logistics. So the space where you can situate between hidden spoke about raw materials, more like in processed materials, but those that you can use for other applications, specifically sources for food. We were talking barley specifically. So he was mentioning that the shorter logistics basically achieves sustainability because that's again social, economic, everything you're getting in here is you cut on that, cut on emissions, and also cut on costs. So
Yeah.
that's huge. I think it's right because different views, right? Different people from different perspective, different industries. Even he's now in food manufacturing. We're talking
Yeah.
here vaccines and the model, but they very much aligned. The thought process is the same. Transportation is huge logistics, but
Hmm.
nevertheless, it's important to have similar conclusions because that can be addressed more constructively. And I think,
Exactly.
yeah, whatever you brought about the chain, the analysis of cold chain vaccines, especially for vaccines, it resonates big time because we we don't have for many formulation which is avoiding cold chain. Most of them
Yeah.
are sitting in this realm. Cold chain is a
Hmm.
mandatory part, such as formulation, but at
Hmm.
the same time, it constitutes certain issues. And also want to highlight, I know we'll have only a few minutes left, but when you were talking about nano DSF, I want to highlight that waste produced by that instrumentation is really small, meaning these capillaries are
Yeah.
tiny, and
Hmm.
none of this goes into landfill because it's all for incineration. So realistically, it's very from that perspective also, from sustainability perspective,
Yeah,
it's a
yeah,
good
absolutely. Even
to
TRPS, because
same,
in
yeah.
the initially we mentioned and development phase, first we need the minimum testing required and the minimum volume of testing required. So with TRPS uses such a small volume of sample, and it gives you a lot of information about the particle sizing. So that's another thing.
Yes, fewer material, fewer. It's from the techniques perspective as well, like a different technique for different purposes. Then this is
Yeah,
great, but also some of them you can highlight that okay, those are actually also contribute to sustainability because fewer materials used or waste generated is it's a small amount of waste. So
yeah,
if it's either eliminated or reduced, I think in this case we can say reduced
yeah.
significantly in terms of testing. I'm just vigilant about time. We only have
Okay.
really 3 minutes. If you want to, say something on a final statement, especially viewers, would be great.
I think again, it's a very great platform to come and share some of my insights about what we do and like because some of these techniques we use on day-to-day life, and but thinking from a sustainability point of view, I think it's very important because we don't usually think in that perspective that okay, a lot of I mean the whole idea is that okay, minimum testing is okay, but minimum test volume is fine, but in terms of consumables, it's sometimes in the research we don't think in that way that okay, the whole idea is more to about the technique and the potential of the technique. But thinking from a sustainability point of view, it's very important that when we're doing this, it's also this can also be in the consideration that the minimum waste to be generated or minimum consumables to be needed, so that techniques can also be given priority from sustainability aspects.
Thank you so much. Thank you so much for coming here today, Rahul, and for the
Thank you so much.
presentation, touching
Yeah,
so many topics, something like, familiar, dear to the heart, and some really good giving perspective to, how we're moving forward collectively and, touching on sustainability. Thank you so much. Great pleasure to
thank
see you here. I hope, we can touch base and reconnect and I'll see more presentations from you on this channel. Dear
sure. Thank
viewers,
you.
thank you so much. Yes. We'll see each other shortly. We'll be having new videos next week. So bye for now. And thanks to Dr. Rahul again. Bye.
Thank you.